Hi, hello my dear students. Welcome to our channel Rajesh Biology and I am Rajesh, MSc Zoology. Today our topic is implantation. So I just want to introduce a few new terminology related to this implantation. Before that, you should know the difference between the developmental biology and the embryology. Developmental biology means from the birth, okay, like one baby girl or a baby boy is born and his growth, the birth, the next growth, and the puberty, the changes, hormonal changes, physical changes, behavioral changes at the time of the puberty, and from the puberty up to the reproduction and the copulation, insemination, fertilization, implantation, embryonic development, fetal development, pacturation, that means delivery, lactation, and he become developed. So entire life cycle, the cyclical changes of the any organism, reproductive cycle, together called the developmental biology. Okay, in that developmental biology, one sub track, one sub branch, okay, a particular period of time is called embryology. So embryology is the study of developmental changes in the embryo, in the zygote. Okay, up to when? Embryology is a sub branch under developmental biology, and in this branch we are going to discuss about the development which takes place within the zygote up to it becomes a fetus. Okay, how many days it takes place to become the fetus? Okay, 8 weeks. Up to the 8 weeks, okay, it is called embryo. From the ninth week, it is called fetus. 8 weeks it is called what? Embryo, and from the ninth week it is called fetus. So all the major changes takes place in the embryonic stage only. Like heart is formed in the embryonic stage, the notochord, and the nervous system is developed, and the limbs are also formed. Okay, everything takes place in the embryonic stage. Okay, once all the major organs are formed, from the third month it is called fetus. Okay, so now I am going to discuss about in this stage, embryonic developmental stage. In that embryology also a particular phase that is called implantation. Okay, we already completed the fertilization. From fertilization up to the implantation, the changes which takes place within the zygote, that is I am going to discuss in this class. Okay, now just come back what we discussed. Here it is the zona pellucida, a glycoprotein layer which protects the secondary oocyte at the time of ovulation and which protects the syncaryon after the fertilization. This is the zona pellucida, a glycoprotein layer. Okay, corona radiata is going to gradually disappear. The sperm receptors are also digested by the cortical granules. Everything we already discussed. Okay, within this there is a zygote. Zygote means in this nucleus is in a 2n stage. You know, at the time of fertilization we discussed like the male gamete, that means the male nucleus with 23 chromosomes and the female nucleus are fused with one another. At that time only the spindle fibers are formed. For the mitosis, spindle fibers are formed. That spindle fibers captures the chromosomes. Why? If the spindle fibers not capture the chromosomes, all the chromosomes are dispersed. Because there is no nuclear membrane or anything, they just disperse in the cytoplasm. So to prevent the dispersal of the chromosomes, the spindle fibers immediately bind with the baby chromosomes of the sperm and the baby chromosomes of the mother, and they start continue the anaphase of the mitosis. Okay, they can directly enter into the anaphase of the mitosis, and that results in the cleavage. Okay, the next step is the cleavage. Cleavage means what? Generally, cleavage means, okay, if you observe my fist, there is a gap in between the two fingers. You can simply differentiate the two fingers because of this cleavage. So, this is called a cleavage, like that in one cell, cleavage starts, okay, because of this mitosis. So mitosis results in the first cleavage. After fertilization, it nearly takes 36 hours time. 36 hours. That means after fertilization, it takes 1.5 days to form two cell stage. The zygote developed into the two cell stage in this manner, and these two cells are covered by the zona pellucida, and this is going to start moving from ampullary isthmic junction to the uterus through the fallopian tube, like from ampullary isthmic junction to the isthmus, and then in isthmus only it is moving like that in a slow manner. When it is moving towards the uterus, and within this the cleavages are takes place, okay, like this, okay. And then next 3 cell stage, not immediately 4 cell stage is not formed immediately. It is asynchronous division. Asynchronous division means both the cells are not divided immediately at the same time. They take some time, okay. So first 3 cell stage, then it continues the division, it becomes a 4 cell stage. So step by step cell divisions takes place, okay. 4 cell stage, and it becomes again 8 cell stage. 8 becomes a 16 cell stage, like that it is moving. And you should know about few things, like all the cells which resulted in this cleavage, okay, because of the cleavage, the cells which are formed, they are called blastomers. Cells are called what? Blastomers. First point. And all the blastomers are a result of the mitosis, not the meiosis. Mitosis only takes place, okay, the second point. And third one, it is asynchronous division. That means both the two cells are not divided at the same time. First one cell is divided, then the division continues to the next cell. All the 4 cells not divided at the same time. In this 4 also, first it will divide, and then next it will, then next, then this. Like that, it is asynchronous cell division, and that results in the 8 cell stage. Even though it is asynchronous, all the cells are same in size, okay, same in size and shape, and the inner nuclear material also, okay. Not only that, the growth of the cells is not takes place. There is no growth phase in the mitosis. In this mitosis, there is no growth phase. Interphase will complete without the growth phase. So the cells are never grow in size. All the cells are in the equal size. And this, what it is, the outer covering is zona pellucida. It will never change its size. The zona pellucida never increase. Okay, like if we take the balloon, if we send more air into the balloon, automatically the balloon size increases. But here the zona pellucida size is never increases. It remains constant. It remains in the same shape and the same size. Within that only within the ovum, within the zygote, the cleavage takes place. So whenever the cell number increases, the size decreases. The size of the cell decreases, not increases, okay. And not only that, the size of the cell decreases, and all are in the same nuclear material. Cytoplasm is never increases. Cytoplasm is actually distributed to all the cells, but the nuclear material becomes double for every time, and every cell get the equal number of DNA material, equal number of nuclear material, okay. So like this, the 2 cell stage gradually becomes 4, and that gradually becomes 8, and 8 becomes 16. This 16 to 32 cell stage, it look like a mulberry fruit. look like what? A mulberry fruit. So this stage is called morula. This stage is called what? Morula. Morula stage, it look like a mulberry fruit, custard apple. How it look like? Same in that manner it look like. So it is called what? Morula. Mulberry fruit shape. That's why it is called morula. And this morula, in this morula, how many cells are there? 32. 16 to 32 cell stage. And all cells are not attached with one another at this time. So it is look like this. And outer zona pellucida as usual. It is zona pellucida. But corona radiata is already disappear and it is digested at the time of the fertilization only. Only the zona pellucida is there. Okay. So inside the cells are look like this: 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13. It is a round shape, right? So outside, other side there is 14, 15, 16. In this side also 17, 18. Like that we can assume. Okay, it is a round football-like structure. Total 16 to 32 cell stages cells are there. Now it is rearranged. It is rearranged like 2 layers, and inside the cells forms gap junctions. It is outer layer, inside the cells are attached with one another, and that process, the attachment of the mulberry cells or that morula blastomers, the attachment of the cells within the morula, the process is called compaction. By the process of compaction, the adjacent cells are attached with one another, and they adjust and they create a space, and they arrange in 2 layers in this manner. So now it is called blastula. Okay, blastula. Blastula is also don't have much cavity like this. The central cavity maybe it is there, but blastula don't has this great cavity. Once the cells starts the compaction, once the cells get differentiated into layers, that is called blastula. Immediately when it is formed, it leaves some space. It leaves some space. The central cavity, that central cavity or the space is called blastocele. Is called what? Blastocele. And this blastocele filled with the fluid, because the cells are going to secrete some fluids into this. So this is called blastocele. Blastocoelomic fluid, that is the fluid filled cavity called blastocele. And this stage is called blastula. Okay, and formation of cavity, the cavity is formed, right? The formation of cavity is called cavitation. The formation of cavity is called what? Cavitation. 3 words with a somewhat similarity. Capacitation. Capacitation is the biochemical changes of the sperm cell to fertilize the ovum. These changes are happen in the female reproductive tract. Next, compaction. Compaction is the film attachment of the blastomers in the morula. Okay, this is called compaction. And cavitation. Cavitation is the formation of cavity within the blastula. This is called cavitation that results in the blastocele. Okay, it is also called exocoelom. It is also called what? Exocoelom. Primary body cavity. That's why real coelom is called secondary body cavity. Coelom means secondary body cavity, but here it is the primary body cavity. Okay, blastocele. And because of the fluid filled cavity, it looks a cyst. It looks what? Cyst. Cyst means a spherical shape. And outer wall within the cavity is a spherical shape. It look like that. So it is called blastocyst. Mammalian blastula is called blastocyst because of its spherical shape. Because of its spherical shape, the mammalian blastula is also called what? Blastocyst, which is going to implant. At this time, this 16 to 32 cell stage, at this time, it is maximum reaches the uterus. Like if it is the fallopian tube, if it is the fallopian tube, at this moment this blastula is almost reaches here and ready to implant. It is ready to implant. Here the fertilization takes place. Later, because of that ciliary movement, it reaches up to here. Okay, by dividing within the this zona pellucida. At this time also zona pellucida is there. At this time also zona pellucida is present. Okay. Still we didn't discuss the implantation process. I'll erase this, then I'll discuss the implantation process. Okay. Come back to this implantation process. In implantation mainly, what is happening at the time of the implantation? The zona pellucida is going to getting embedded in the wall of the uterus. So this is what I'm going to explain. Once it reaches the uterus wall, so I'll show the uterus wall with the red color lining because it is highly vascular. We already discussed in the menstrual cycle. For this implantation, the endometrium gets ready to become pregnant. Okay, that means endometrium is not becomes pregnant. I'm saying that to hatches or to give the nourishment to the baby, it becomes highly vascular. So it is the thick reddish in color and it is ready to give nourishment, and glandular tissue is also develops. Uterine glands are also develops. Okay, uterine glands are develops and more vascular it becomes, and it is the time to implantation. At this time, the zona pellucida I'm going to show with the yellow color. It is what? Zona pellucida. In this zona pellucida, I'm going to show this embryonic cells like blastomers with the blue color. So this is the blastomers which are arranged in two layers. One is the inner cell mass and another one is outer trophoblast, and in between this there is a cavity. This is called blastocele, and this blastocele is the fluid filled cavity. Now it is hatches out. How it look like? How when a baby chick comes out from the egg. When a baby chick comes out from the egg, how it look like? As like this, it is going to come out from the zona pellucida by rupturing the zona pellucida layer. By rupturing this glycoprotein layer. To rupture, to digest the glycoprotein layer, it should release some enzyme. That enzyme name is trypsin. With the help of the trypsin enzyme, it is going to break down the zona pellucida and it hatches out. Once it hatches out, it look like this. Once it hatches out, now it look like this. And the entire zona pellucida is present outside of this, and gradually it going to disappear. Okay, once it comes out, it is gradually disappear because of the endometrial secretions and other enzymatic actions. Because of the trypsin also it is going to disappear gradually. Now here you should observe two types of cells. Okay, outside cells, that is called trophoblast. It is also called what? Trophoblast, trophectoderm. Trophectoderm. I always used to say to my students every time, wherever you find the word trophozoite stage, feeding stage, okay, adrenocorticotrophic hormone, which is going to stimulate or feeding the cortex to release something. Okay, tropho means feeding, eutrophication, really the lake becomes more nutrients rich, that's why it is going to dead. So wherever you find the word tropho, that is related to the feeding or stimulating nourishment. Trophectoderm or trophoblast, which is going to form the placenta. Actually it is not real placenta, okay, embryonic placenta it is. After fetus is formed, there is a different special placenta is there between the allantoin chorion of the baby and the uterine tissue of the mother. Both are interdigitated to form a structural functional unit of the pregnancy, that is called placenta. We will discuss about that later. But here it is an embryonic placenta, okay, it invades into this, it distributes, it sends its finger-like structures into the uterine wall to hug, to enter, to invade into the uterine wall. This is called trophoblast, or also called trophectoderm. Now come to the inner cells. Outer trophoblast and inner cell mass. Inner cell mass. In this, the junctions are like gap junctions. Outer trophoblast cells are attached with one another with the help of the tight junctions. And the inner cell mass cells are attached with one another with the gap junctions. Tight junctions and gap junctions both are present within this. Okay, now come back. Tight junctions to prevent the leakage of that blastocele or any other nutrients. Inner cells, they want to communicate with one another, that's why inner cell mass contains gap junctions within that for the perfect distribution of the nutrients within the inner cell mass. Actually inner cell mass is going to develop into the embryo proper. It is the embryo proper, embryonic blast, okay, generative or vegetative cell mass it is, vegetative cell mass, okay. Now next what happens, this trophoblast is again divided into two layers. I am going to show here, that is outer syncytial trophoblast, outer what, syncytial trophoblast, and inner cytotrophoblast. Cytotrophoblast means we can observe the trophoblast cells, okay, this is inner cytotrophoblast, but outer all the cells fuse with one another, all the cells fuse with one another, so only one cytoplasm, only one cell membrane, so it is called syncytial trophoblast. Outer syncytial trophoblast and inner cellular cytotrophoblast both are there, and the syncytial trophoblast are invade into this uterine by forming the branches all over, and it forms the villi. It forms what? Villi. It is called trophoblastic villi. It is called what? Trophoblastic villi, okay, which helps to invade into the uterine wall of the mother. Now this process of invasion of the blastocyst into the mother's uterine wall, this process is called implantation. This implantation starts after the 6th day of fertilization. It starts after the 6th day of fertilization to complete all its process, like a trophectoderm divided into outer syncytium, inner cytotrophoblast, and outer syncytium forms some branches, okay, trophoblastic villi to invade into the uterine to get implant completely. It takes 1 week time. So after fertilization, that means after this 1 week, like second week, at the end of the second week, the total implantation process will be completed. This is implantation. I hope you understand in a clear manner. If there is any doubts, you can ask me. And this is about today's class. Thank you. Have a nice day.